thousands upon thousands of skeletal muscle cells to just a few muscle cells here and here. We can count 1, 2, 3 skeletal muscle cells. Here we can count 1, 2, 3, 4, 5 skeletal muscle cells. So what would the connective tissue be in between these cells here? See if you've been paying attention.
Perimysium?
Endomysium. Endomysium. This gray stuff here. So endomysium connective tissue. You can see a nucleus, very, very superficial, just under the cell membrane right there. Nucleus just under the cell membrane. Capillary, running through the endomysium connective tissue right there. Let's see. So longitudinal view, there's a portion of one skeletal muscle cell, 1, 2, 3, 4, 5, bunch of shallow nuclei, absolutely multinucleated. The striations are these right-angle bands that you see, these right-angle bands. And again, this one doesn't actually, except for the endomysium seen end on. That's the only place on the model where endomysium is shown. Now, going in the other direction, from deep to superficial, endomysium. Then we've got perimysium wrapping these 5 cells. Notice it does not actually go between the cells; it stays on the outside. So it folds around, does not go in between. Folds around, it's wrapping the outside of these 5 cells. So the perimysium around the 5-cell fascicle or cell bundle. Next layer out would be epimysium. This would wrap all of the fascicles of the entire muscle, epimysium. There should be one more layer on top of this. There should be fascia on top of this, which they are not showing you. Now, what else? So again, we've got these capillaries that do run through the endomysium, not shown on this surface view. Here, we've got something interesting. That's a tendon. So right there, where muscle meets tendon, muscle-tendon junction. Remember, tendons join bones to skeletal muscles. So if you follow that to the left, we would be going to a bone. Let's see, what else? So the cell membrane of a muscle cell is called the sarcolemma. Sarcolemma, 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 sarcolemma. And each skeletal muscle cell sarcolemma has a region shown in gray here. This is sarcolemma, shown in gray, has a specialized region of the sarcolemma where all of the neurotransmitter receptors are. So you guys, have you learned much about muscle in lecture yet?
No.
Okay, so all right, but you've done nervous. So a somatic motor neuron, which innervates skeletal muscle, secretes what neurotransmitter?
Norepinephrine.
Now, somatic motor neuron.
Acetylcholine.
Skeletal muscle. Acetylcholine. Have you guys learned cholinergic receptors?
Yes.
Okay, good. Have you learned nicotinic versus muscarinic? Okay, so you guys tell me. This has got to be what kind of a neuron? I've kind of been laboring it here, so what kind of neuron must this be? Somatic motor neuron, somatic efferent neuron. When it fires an act potential, what will it be secreting from its axon terminals? Acetylcholine. Onto the motor end plate portion of the sarcolemma, which has what type of acetylcholine receptors? Nicotinic cholinergic receptors. Okay? What do we know about nicotinic cholinergic receptors?
Excitatory.
They're always excitatory. Do you know why they're always excitatory? Do you know what they are structurally? They are ligand-gated sodium ion channels. I said, oh, right, we did learn that, correct? Okay. So why would that always be excitatory? Acetylcholine binds to the nicotinic cholinergic receptors. The ligand is the acetylcholine. The gates open. What happens?
Sodium influx.
Net movement of sodium in, facilitated diffusion through the open gated sodium channel, net movement of sodium in. And what does the gain of positive ions do to the cell's voltage?
It decreases it to 0.
Okay, so it actually makes it less negative. So it actually moves it toward the positive. Yeah. So we're talking about the resting membrane potential of minus 70 millivolts. when positive ions are coming in, it's going to go from minus 70 to minus 65 to minus 60 to minus 55. So that's depolarization. And depolarization is always excitatory. So nicotinic cholinergic receptors are always excitatory for those reasons. Now, so that's got to be the distal end of a somatic motor neuron, somatic efferent neuron. The axon terminal is lined up over the motor end plate region of the sarcolemma of this particular cell. Let's see. And inside each of these cells are log-like objects called. So if the cell itself is also known as a myofiber, inside each myofiber are many, many log-like objects called myofibrils. So this is one myofibril here. If you look at it structurally, it consists of alternating A bands and I bands. This is complete I band right there, from there to there. Here's another complete I band, and the center of an I band is a Z disc. There's another Z disc right there. This is a complete A band. So you've got I, A, I, A, I, A, I. So each myofibril is an alternating series of A bands and I bands. And again, the parts are also important to know. So the A band from there to there has a lighter region, although they did not make it very light on this model, has a lighter central region called the H zone. From the German word helle, meaning light, literally meaning light, lighter central region. So the H zone. And the center of the H zone, which is also the center of the A band, is an M line. So there's an M line right there. That's from the German word Mittel, which literally means the middle of the A band, or the middle of the H zone. So I band with Z disc, A band with H zone and M line, I band with Z disc, and so on and so on and so on. Now another term on your objectives handout is the sarcomere. The sarcomere is the smallest contractile unit of any muscle cell. So when a muscle cell is contracting, in reality, all the individual sarcomeres are contracting like microscopic accordions. So the myofiber here does show a sarcomere. From Z disc to Z disc is one sarcomere right there. From there over to there, that is one complete sarcomere. From Z disc to Z disc of a myofibril is one complete sarcomere. So there are two ways to look at the structure of a myofibril: as a series of alternating A and I bands, or as a series of connected sarcomeres. Sarcomere, 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 sarcomere. Either one of those is an accurate way of looking at the structure of a myofibril. But so this then is the actual contracting part of the cell. So there are hundreds of thousands of them within the cell, all contracting at the same time when the cell is scrunching up. Any questions about this group of skeletal muscle cells? Anything at all? Okay. We've got a few more skeletal muscle cells here.